Before the main event today, I've been allowed to make a small presentation about the next International Mycological Congress, which will take place about a year from now in Bangkok, Thailand. And if you want to learn about it, the simple thing to do is just Google IMC for International Mycological Congress 10. You might have to add Bangkok, and then you'll get the website. The steering committee is chaired by Lekha Manak and Morakot in Thailand, and includes a number of Thais and Nick Reed from our part of the world. And here's Lekha with some ice cream. And there are going to be seven types of sessions: environment and ecology, evolution and systematics, diversity and conservation, genomics, genetics, cell biology, pathogenesis, and biotechnology. And in addition to me, there are several people at the meeting, Pedro, Dominic, Keith, and David, who are involved in organizing the program for Bangkok in 2014, and you can please talk to them if you have questions. Now, in addition to the meeting, there's some interesting things in Thailand from a mycological perspective, including the largest herbivore dung you'll ever see. And if there's large herbivore dung, there have to be large herbivores. And Delia and I actually saw these animals in the wild on our last trip to Thailand. And this little one said, "To see you at IMC 10. So I want you to remember a year from now in Bangkok, and that's how you can find out more about it. And now it's my pleasure to introduce Carrie O'Donnell.
Thanks everyone for showing up for the main show this afternoon. Before I do the introduction, I wanted to express how much fun MSA had interacting with Mike, the AMB planning board at APS, and last but not least, Betty Ford and her colleagues in St. Paul. So they all worked enormously hard behind the scenes to make this happen, and I think this is appropriate that I acknowledge what wonderful collegial interactions we had over the course of the planning. I also should acknowledge the John Karling committee, none of whom could be here, but it was chaired by Cameron Curry at Wisconsin, Ann Pringle at Harvard, and Joe Heitman at Duke Medical School also made the selection for Professor Howlett. A brief introduction to her academic background. She took her bachelor's and PhD at the University of Melbourne, Australia's leading academic institute, and certainly one of the top academic centers in the world. After her formal training, she did postgraduate studies at UC Berkeley and Harvard, and Stanford, and it was in David Perkins' lab at Stanford where she developed the genetic and molecular genetic skills that have enabled her to develop the system study in the genetics and genomics of the blackleg disease of canola caused by Leptosphaeria maculans. And probably most of you know that this disease threatens canola production worldwide. Without any further ado, I'll turn the podium over to you, Barbara. Thanks a lot.
It's wonderful to be here, and I'm very, very grateful for this honour, and I thank the committee. So today I'm going to talk to you about evolution of virulence in fungal pathogens of plants, and I'm going to talk about work that we do in the laboratory. This is a School of Botany, and in the field. And this work is done in collaboration with my very valued colleagues below, and I will mention them as I go through the talk. Now, we must remember that plant pathogenic fungi or plant pathogens co-evolve with their hosts. And to be a pathogen, they'll overcome resistance responses. And the virulence factors that I'll discuss today are effectors. So they're small molecules or proteins that can facilitate infection or indeed induce host defence responses. Sophie Kamoun wrote a very nice review of a colleague last year in Nature Review Microbiology, where he considered what were the properties of fungi in terms of evolution of virulence. And these are some of the things that he came up with, and they're the themes that will come from throughout the talk. So the reproduction systems, clearly fungi with sexual reproduction systems have the ability to shuffle effectors, molecules around. When we look at genome structure, as more and more fungal genomes are sequenced, we find that some of them have very large genomes, and they're comprised mainly of transposable elements. The affected and genes are distributed throughout them, and there must be some sort of trade-off between maintaining such a large genome and yet success as a pathogen. Other features of genome structure are when you get horizontal gene or chromosome transfer. A very nice example of this is that just one protein, tox-A, from Staganospinodorum, a pathogen of wheat, transferred to Pyranoptera triticirrhopentis, which is the tan spot fungus, in the late 40s, and made this a much more virulent pathogen. This is the work of Richard Oliver's lab and Tim Friesen's lab. And also we know that horizontal chromosome transfer is very important. Fusarium oxysporum has lineage-specific chromosomes that are responsible for host specificity. Mutation, point mutation, deletions are also important. Now I'm going to bring these out, or try to bring this out, in the work that I'll tell you about today on blackleg of canola, which is caused by the dithidiomycete pathogen Leptosphaeria maculans. This is an important crop worldwide. In Australia, it's grown in rotation with cereals. It confers a 15% yield benefit on subsequent cereal crops because of the glucosinolates in the root that take all fungi in the soil. The edible oil is 42%, and it's used for industrial purposes, biofuel, but it's edible as well. It's not a big crop in your country, but in our country, we last year had 2.4 million hectares. In Canada, it's a very important crop, and these are the things that you would see in the supermarket. Now, the major threat to this industry worldwide is the fungus Leptosphaeria maculans, the disease blackleg. So if we start with the stubble, at the end of summer, in Australia we grow minimum tillage, so we don't cultivate the soil. You get loads and loads of pseudothecia that release wind-borne ascospores. The ascospores germinate, penetrate the stomatal aperture. You can see on the young seedlings, you can see that they're infected. These are the asexual pycnidia. It's called blackleg, the disease, because the fungus, the hyphae, grows right down to the crown of the plant, the leg of the plant, and you get a blackened canker, hence the name. Indeed, it keeps going right down to the roots. So you have the plants lodge, loss of yield, and then crossing occurs over summer and the cycle continues. So a farmer's pretty pleased if just before harvest, this is what the crop looks like. Sorry. But at the seedling level, you can see here the pycnidia on the cotyledons. And at the end of the season, if there's been a lot of disease, then you'll have a lot of stubble with, that's blackleg infested. And you can see it here. Now, we grow canola in Australia over a long period of time, about 7 months. High intensity, I mean, you can see a field, a landscape of yellow. This is on the Eyre Peninsula. And it's a temperate climate. So it's ideal situation for sexual crossing, and there's lots of stubble. So, for instance, this year we have a large amount of stubble inoculum. We've had a fair bit of rain, so there's high disease pressure. So it's controlled by rotations with cereals and pulses, so that the pseudothecia, the ascospores break down and the stubble breaks down over a couple of years. Fungicides now beginning to be applied to seed, fertiliser and sprayed, often unnecessarily. But the most important strategy has been resistance breeding. Single genes at the seedling stage and also minor genes at the adult stage. So we can see here this has no seedling resistance. The cotyledons have been infected and the fungus has grown right down. This is at the cross-section of the base of the stem, a lot of disease. This has seedling resistance, and so there's no disease either in the cotyledon or in the stem. This has adult resistance. It doesn't have seedling resistance, but the hyphae are stopped as they grow down. So without all of these strategies, we would lose about $330 million per year. So it really is a problem for us. This is a gene-for-gene interaction at the seedling level. And so we've had 10 seedling resistance genes mapped. One indeed has been cloned by people in Ag Canada. And the complementary virulence genes have been mapped in the fungus, and 5 of these have been cloned. Now, throughout the talk, I'll refer to our colleagues led by Tilly Ruxell at Internet, because he's really been a major driver in this field. And these virulence genes are typical effector genes. They're small, secreted, cysteine-rich proteins. So here we have the name of the virulence gene, the corresponding brassica resistance gene in the plant, in Brassica napus, in this case Brassica juncea, napus, Brassica rapus. The brassicas are very closely related. So this fungus, Leptosphaeria maculans, is a high-risk pathogen for overcoming disease resistance. You get prolific asexual and sexual reproduction on stubble, and they're wind-borne large populations. So when you have extensive sowing of a cultivar with major gene resistance, it confers strong selection pressure on that resistance gene, and so the frequency of virulent isolates in the population increases rapidly. I'm going to tell you about 2 situations in one geographic part of Australia where resistance was overcome in 2003. We learned from it and were able last year to avert a similar situation. So this is the map of Australia, and I live in Melbourne. This is the Eyre Peninsula. It's an area that there's a lot of canola grown. You saw a picture of it before. There's not much pulses, so there tends to be cereals canola. And in 2000, a cultivar was released there with major gene resistance, 2 resistance genes. It was sown extensively, and the cultivars looked superb. We couldn't even see cotyledonary lesions, so the growers really were pretty impressed. So this is it in 2001 and 2002 on the left. You can see being cut for threshing. But in 2003, completely wiped out. Had a PhD student, Stephen Marcroft, here he is, in 2003, in a field plot, and here he is 50 metres away, and with a different variety. And you can see this is very specific, this breakdown. Only this is broken down. This resistance gene's fine. Now, there were severe yield losses on the Eyre Peninsula, and the seed was withdrawn from sale. And here's Steve in the local paper, and we had a lot of press about lots of good puns and headlines about it. And you can see that it was very important because it's an Aussie magazine, and yet the beer award is much smaller in the background. And believe me, Steve knows a lot about beer. And I cannot move this. Yes, I can? Good. So this is what we saw in 2004 because, of course, we kept the field sites going. One line next to another, you can see that we're still seeing severe breakdown. I can't move this. Now I can. And yet, when we tracked it over a number of years, we could see 2002, 2003 with the breakdown. By 2006, we're beginning to see in our trials that we're not having so much disease. But if we track the other variety, we can see that when it'd been sown for quite a while before this one was released, and so we can see quite a lot of disease, but when this was released, this one wasn't sown much, not much disease, and here again we're seeing quite a bit of disease. So, it's a breakdown of resistance, and it was an increased frequency of isolates attacking cultivars with the resistant. It wasn't a new strain or anything else because we had isolates in the lab that we'd collected 15 years previously that could cause disease if we did glasshouse trials with them. And also it broke down in other areas where this variety was grown extensively. So it's a boom and bust situation. So we had some questions. Why does virulence evolve so quickly in populations of this fungus? What are the changes in the virulence gene ABRLM1 that are responsible for the breakdown? And can the risk of breakdown be reduced through monitoring the changes of resistance, of virulence? So at the same time, we were working with Leptosphaeria in the lab, and it grows on defined media. It's quite a lovely fungus in many ways. You can do sexual crossing. You can inoculate plants, you can transform. Gene knockout's very difficult, but we do gene silencing. You can separate the chromosomes. So you can see it here. And in terms of genome structure, we were very surprised. Kim Plummer in 1993 noticed that when we ran pulsed field gels, the isolates had different sized chromosomes, and this had not been seen previously in a plant pathogenic fungi. It enabled us to do a couple of things at least, to add up the sizes of the bands and work out how many chromosomes there were, what the genome size was, and we calculated about 38 megabases. But we also noticed that during crosses, they could cross, because we saw prolific sexual reproduction, we could cross them in the lab, they have different sized homologs compared to the parents. So on this one we can see that the progeny in the tetrad have the same size as the parents, but there are recombinants here in size. So even though the chromosomes were quite different in size, we could still get crossing. And we believe that these chromosomal length polymorphisms were due to different amounts of repetitive DNA on the homologs. Now, at that time, we hadn't done any, we really didn't know that. That was just a guess. And so slippage during meiosis would generate the length polymorphism. And indeed, when we and others sequenced repetitive DNAs, we showed that they were transposon derived. So we knew at that stage that the genome structure, there was repetitive DNA, and also that we had chromosomal length polymorphisms. And the next thing that we discovered was that this fungus undergoes repeat-induced point mutation. This occurs in many Ascomycetes and indeed has been characterised really by pioneering work by Eric Selker's group in Oregon. It mutates multi-copy DNA, and it occurs in haploid nuclei after fertilisation prior to meiosis. So you get transitions, C, G to TA, nucleotide base pairs, and this generates stop codons and hence can inactivate genes. So that you can use bioinformatics to pick up RIP signatures because the mutated CGs are usually followed by AT. And Alex Idnerm discovered this by chance. He had looked at he was generating a bank of what were called REMI insertional mutants of Leptosphaeria maculans using hygromycin resistance as a marker. And when he had a non-pathogenic one, and it had some tandem copies of hygromycin resistance in it, he crossed it to a wild type to make sure that the hygromycin and the phenotype co-segregated, but he found that everything was hygromycin sensitive. So when he sequenced these these isolates of hygromycin sensitive, he found that the hygromycin gene was there, but there was extensive RIP mutations. So a few years later, in 2007, we had the opportunity with Thierry Rouxel and Mylène Bellaston that the French sequencing agency Genoscope agreed to sequence the fungus. And 12,500 genes, similar to most ascomycetes, but it was a surprise because the genome size was so large, 45 megabases compared to a very closely related dithydiomycete, Staganospora odorum, with 37. There was repetitive DNA, and that indeed was responsible for that large size difference, 35%, whereas it's only 9% in Staganospora. And this repetitive DNA comprised class 1 retrotransposons, and these are 83% of the elements, and class 2 terminal inverted repeat DNA transposons. We saw, as we would have expected, repeat-induced point mutation, and it had a unique genome structure. Maybe I could get another slide changer. No, I've got it. More quickly than I hoped. So it had the unique genome structure is that it's a patchwork genome. There were gene-rich GC regions and gene-poor repeat-rich AT-rich regions, isocores. So if we look at the, this is the GC content, and this is very gene-dense, one gene, 2.4 kb. And yet, if we look here, we see in the black, the white here are the genes, the very little white there. This is very gene-poor, repeat-rich blocks. And there's one gene per 30 kilobase. They're degenerated transposons. And they have only 3.5% of the genes in the genome, but 20% of the effectors, the small secreted proteins. So it's an invaded plastic genome. It's readily, where genes are readily gained, lost or mutated. So we were really thrilled when this was published. And you can see it's the next, this is the Melbourne team. We're in University House pretty early in the morning, and we're on to our second bottle of wine. But we're very pleased because genome papers, as many of you know, are hard to deliver and achieve. As is moving the next slide. Got it. So, why are the genes in these regions so amenable to mutation? Well, if you consider the lining up of, we've got here in the blue an virulence gene. So during meiosis, if you get the repeats lining up correctly, then you'll get virulence genes that are identical in parents and progeny. But if you get slippage, then you'll get you'll get two copies in this progeny. Now, if they're close together, you're likely to get RIP, so you're getting gene duplication, and you'll get gene deletion. So it's a very plastic region of the genome to have such genes. So we wanted to know, so we then knew why the virulence evolved so quickly, because the virulence genes are located in these repetitive regions where RIP mutation and gene loss can occur readily. What were the changes in the virulence genes that conferred the breakdown of the sylvestris resistance? Karling Cell had shown that this particular virulence gene was in a 530 kb region, AT rich and low recombination. But it was also flanked by three other small secreted proteins, and you can see I've put red loops around them. Here they are. And there were several non-coding, non-repetitive regions as well. Now, the red background are the truncated or the inactivated transposable elements, highly repetitive. We'd been collecting isolates from the field in the Eyre Peninsula for many years, so we had a look at 295, 137 before the breakdown, 158 after the breakdown. And this is what we found for these four effectors. With the one that we were most interested in, AVRLM1, the mutations were only conferred by deletions. So that was the only mutation type of deletion that conferred virulence. And there was an 8-fold increase in these deletion alleles after the breakdown. But we were extremely surprised to see that another virulence gene, there was a 6-fold increase post-sylvestris breakdown, and yet there were no cultivars with sylvestris with the corresponding resistance gene sown there. So there was no selection pressure conferred by any RLM6 cultivars. And furthermore, there was RIP in repeat-induced point mutations in the RLM6 and also in one of the other effectors. And indeed, of the 13 alleles that we found, 7 of them showed RIP. There was no RIP in any of the isolates that we'd collected before the breakdown. So we're pretty intrigued about this. So we then decided with James Hayne and Richard Oliver from Curtin University in Perth to have a look at the direction of the RIP mutations with respect to their proximity to the transposable elements. Because remember, previously, RIP had only been shown to occur in multi-copy genes. So the red shows a high level of RIP, over 50% of the sites eligible for RIP, and then this shows that from the 3' exons down to the 5' UTR, there's a decreased gradient in the amount of RIP that you see. And similarly, when we had a look at the genes in that particular area, the one with the highest number of alleles was one of the non-coding regions, just adjacent to the transposable elements, and then AVRLM6, and then the other coding genes. So if we have a look again at this region, we can see the genes that I've just told you about. We can see the AVRLM6, we can see the non-coding region, and we can see that there are RIP gradients. So in other words, as the part of the gene is further away from the transposable elements, then you're seeing less RIP. So this was the first study to look at linked effector genes, and they evolved differently depending on which particular gene they were. We saw repeat-induced point mutations, we saw deletions, and we saw amino acid substitutions. We also, as I said, saw RIP mutation in single copy genes. And we hypothesise, and again, Karling Cell has seen something similar, that it's leakage from the transposable elements. We also saw that the frequency of isolates with virulence alleles of the resistance gene AVRLM1, which was complementary to the resistance gene that was being sown, a linked or closely, an avirulence close by increased dramatically after extensive sowing of the sylvestris resistance. Now these genes are in a low recombination, an area of low recombination, and so we, with Bruce McDonald and Patrick Bruns from Etihad, Switzerland, we looked at this and concluded that it was a selective sweep whereby selection at one locus affects the frequency of virulence alleles at a linked locus, and this is called hitchhiking. So, the implication for this, of course, for farmers is that if they're growing a variety, then they need to know where the avirulence genes are in the pathogen. So selection imposed by widespread deployment of a resistance gene can affect evolution of a complementary avirulence gene and the closely linked one. So as well as for plant breeders to have knowledge of the plant genome, it's also important to have knowledge of the fungal genome. So to maximise effective resistance, a lifetime of resistance genes, each resistance genes cost in Australia at least $4 million to commercialise. So we need to come up, our research is funded by farmers, we need to come up with a way to get durable resistance. So we know that virulence frequency of fungal populations can be manipulated by sowing cultivars of different resistance genes. We saw that on the Eyre Peninsula in the field stuff, and we've also done plot studies to show that if we rotate resistance genes, then we put less pressure and we get less disease. So we then set about to ask, well, can the risk of breakdown of resistance be reduced by monitoring the frequency of virulence in fungal populations? And so I lead a national program, it's called Staying Ahead of Blackleg. It's a deliberate title because we're really telling farmers, and I think they get the message, we're not going to beat the disease, we've got to stay one step ahead of it. It's farmer-funded research. They have 1% of their farm gate value, goes to the Grains Research and Development Corporation, and then it's contracted out to people like us to do both the field work and the lab work. So we have people in all the regions. I'm in Melbourne with Phil Salisbury and Angela Vanderwaal, who's also doing a lot of field work. So she's up in Horsham in the canola growing region. We've got Andrew Ware in the Eyre Peninsula. We've got someone, Kurt Lindbeck, in New South Wales and in Western Australia. So this is sort of the canola growing area. So, firstly, if we're going to rotate resistance genes, we need to know what resistance genes we have in our lines and cultivars already, and surprisingly enough we don't, or the breeding companies don't. So we've spent a lot of time developing just a differential, very classic stuff, differential set of isolates that we use to, with some known standards, to put all our lines into particular resistance groups. So here you can see susceptible resistance, and then this is a glass house full of adult plants that have been cut off at the stem, and Angela's scoring them for adult plant resistance. And then they're put in particular groups based on the complement of resistance genes. We have 36 sites across the country in the canola growing regions, and at harvest we do the internal stem infections and grade how much disease there is. We also take stubble, the trash, back to the lab and culture single ascospores from the pseudothecia, do glasshouse trials where we inoculate on different cultivars and we assess disease. But of course, that's pretty time consuming. So we've developed a way of looking at fungal populations, and that's when we put the stubble of the different cultivars above seedlings of different cultivars also. The ascospores are released onto the seedlings below, and then we assess the plant, the disease maturity. So it's very high throughput. And this is an example here of where we've got a variety garnet, and if we put garnet stubble on top of it, then the ascospores that are released onto it will grow down and infect it and it'll die. But if we put stubble from another cultivar, they look perfectly healthy. So this is a way of taking the stubble from all around the country and working out the propensity of that stubble to cause disease. And that, of course, is next year's inoculum. And we've also developed a molecular assay to assess the virulence alleles. The stubble's put in a wind tunnel, the ascospores are captured on tape, and the DNA is extracted and analysed by quantitative PCR and we look for presence and absence alleles of AVRLM1 and AVRLM6, and in relation to how much, we can work out how many spores there are by the amount of the ITS region. We've got another virulence gene where there's a virulence conferred by a single nucleotide polymorphism, and Angela van der Waals developed a pyrosequencing assay. So we then take the stubble, we measure the frequency of these virulence alleles, and we can determine in the region from which we got the stubble whether resistance may break down, and then we advise farmers on cultivar choice. So getting the message out there, well, the Grains Research and Development Corporation developed these disease management guides. There are regional disease warnings. We go to field days, and we encourage farmers to survey their own crops, so to get a pair of secateurs and have a look at each year and see what's going on with respect to the propensity for next year to have disease. These are, again, Steve Marcroft, 12 years later, looks pretty much the same. Angela van der Waals, a postdoc in my lab, and Andrew Ware, who's on the Eyre Peninsula. They're going around looking at these field trials, and you can see they've cut the base of the stem and they're assessing disease. So they noticed in 2011 that although there was no yield loss, there was a lot of blackening of stems in this particular variety, Hyola 50, and when they did the test with the stubble above pots of the seed, they could see that all the Hyola 50 plants died. This is a positive control, whereas another cultivar with different resistance genes didn't. So in February, we released a warning to farmers on the Eyre Peninsula saying this is what the change in infection, cross-section infection we've seen just in 1 year. So don't plant this variety, and we told them which ones that they should indeed plant. And because it's farmer funded, they took our advice, and so there was very little of that seed sown, but of course we sowed it in the field trials. And the varieties that we told them to sow, which is these ones and these ones, they're very little disease, but about 90% cross-section infection there. So if they'd have planted it, it really would have been bad news. Now that, of course, is just on the Eyre Peninsula. This is very regional. But here it is. You can see that the cultivars look pretty terrible in our field trials, whereas the other ones don't, where the resistance hasn't broken down. And yet the cultivars, Hyola 50, the Group D ones at Wagga, which is in New South Wales, look terrific. So this hit the local press with a Bob Dylan quote, "The farmer's answers are blowing in the wind." It should have been the ascospores are blowing in the wind, but any press is good press, so we weren't too unhappy. And we calculate that if there are about 60,000 hectares sown to canola in 2012 in that region, and based on the price of canola per tonne, etc., we reckon that they've been saved about $18 million. But the other good news about it was that the seed companies didn't take the varieties all from Australia, they could sow them elsewhere. So it was a sort of a win-win for the farmers and the companies. Now, Leptosphaeria maculans is a very aggressive pathogen, but it's part of the species complex. And this species complex includes, Leptosphaeria biglobosa, which is not as aggressive, but it's an important pathogen in France. And in 2005, Kirsten Voigt and we put together this based on sequencing 3 genes. We're able to look at the relationships. We can see Leptosphaeria maculans lepidi, extremely similar, but it's a pathogen of Lepidium, a cruciferous weed. It doesn't cause disease on canola, and we also have these other ones. So we're quite interested to see why is this such an aggressive pathogen. You can see if you infect with Leptosphaeria maculans brassicae, the cotyledonary lesions, and then the death of the plant. However, with the biglobosa, you get big cotyledonary lesions, but the fungus is stopped, doesn't get down to the base of the stem. So we have a comparative genome project, and this involves Eukaryotic Cell's group, particularly a graduate student, Jonathan Grandobert, Conrad Schock from NCBI, and Hossein Borhan from Ag Canada. And so lots of questions. When did these Leptosphaeria species diverge from each other? Are the other Leptosphaeria species transposon rich? When did the transposable elements invade? How prevalent is RIP? And how was this isochore genome structure developed, where you've got the AT-rich blocks and the GC-rich blocks? Gene synteny, I mean, you imagine all of the questions that we've got, but I guess the main one down the bottom, why is Leptosphaeria maculans brassicae such an aggressive pathogen? So at Eukaryotic Cell's group sequenced 4 of these. So we'd already got the Sanger sequence of the Leptosphaeria maculans brassicae. This is Leptosphaeria maculans lepidi, Leptosphaeria biglobosa thalaspi, and Leptosphaeria biglobosa brassicae. So this is quite a good pathogen. And we sequenced Leptosphaeria brassica canadensis at the University of Melbourne. Now this is next generation sequencing. This is 554 and Illumina sequencing. This was just Illumina sequencing. But nonetheless, first thing to notice is that the genomes are much smaller, and indeed that can be accounted by the high amount of the transposable elements in Leptosphaeria maculans brassicae. The gene numbers are sort of pretty similar, and Leptosphaeria maculans brassicae 650 small secreted proteins, but the others, I mean, this is a weak pathogen, but it's got 737, etc. So a similar order of magnitude. One of the interesting things is, these are preliminary results, but when we compare, align the genomes of these two, remember they're very closely related, you see synteny, gene synteny, same order, or perhaps mesosynteny, we've got a bit of a shuffling within the chromosomes, but wherever there is a inversion or whatever, it's always interrupted by a transposable element, and there's RIP mutation throughout all these genomes. So we're trying to answer quite a lot of questions. We're also doing RNA-seq to have a look at which of these secreted proteins are expressed highly. But just one snapshot that we're interested in is species divergent and transposon invasion. So this is Conrad Schock sequenced 19 proteins, or he aligned the sequences of 19 proteins for about 51 taxa, and this is just the Leptosphaeria complex, but he did a lot of Dithydiomycetes as well. I'm really just showing you here, there are 41 transposon families, and so there are two transposable elements that are in other Dithydiomycetes that are not Pleospirales, and you can see across here that we're getting ones that are specific to each particular branch, etc. But about 3 to 5 million years ago, we had this massive invasion of 4 families of transposable elements that aren't seen in the other genomes, about the same time as we got divergence between Leptosphaeria maculans brassicae and Leptosphaeria maculans lepidae. So there's lots to do, as you can imagine, with this. So in summary, I've told you that Leptosphaeria maculans is a high-risk pathogen for breakdown of resistance in canola cultivars. And going back to the theme of the talk about the evolution of virulence, well, the evolution of virulence in this fungus can be accounted by a number of factors. Firstly, prolific sexual reproduction, and also a patchwork isochore fungal genome with the avirulence genes embedded in repetitive regions that have these transposable elements that have been inactivated by RIP mutation. And the RIP mutation itself can leak into single copy genes, and so most of the single small secreted proteins that we've been characterised are single copy genes. We also saw something quite interesting, that selection imposed by widespread deployment of a resistance gene can affect evolution of a complementary avirulence gene, which we would expect, but also that of a closely linked avirulence gene. And so we monitor this race-specific virulence of populations across the country derived from stubble. We do it both through the spore trap assays, the wind tunnel assays, and also in the glasshouse and in the field. And we're able to avert a breakdown of resistance in this region of Eyre Peninsula last year. And we're very excited about what is going to be coming out from the comparative genomic study of Leptosphaeria maculans that we're doing at the moment. So thank you very much for your attention. If anyone wants to ask a question, I can't see them, but I could answer. I can't see anything. Oh, thank you.
Hello? Hello? Okay. So if you're just trying to see the causal and effect relationship, is a transposable element is driving the generating the new effector, or is a result?
Is a transposable element driving the presence of new effectors? Is that the question? Yes? Yes. We don't really know. I mean, we have situations where we find a lot of polymorphism in infectors, and then we have situations where we either have it there or there's a deletion of it. We know that when the transposable elements invade, they're sort of nested. They invade where they've been previously, but I guess we just don't know. But we do know that we've never seen an allele of an avirulence gene that is conferred by RIP that has specificity. RIP introduces, sure, amino acid changes, but it introduces stop codons prolifically.
Is there any evidence with the different genomes now that horizontal gene transfer is playing a role in virulence adaptation also?
Is there,
given
I
the
don't
species,
hear well.
I'm sorry, given the species complex you have
Yes.
here, and now the different genomes that you have,
Yes.
comparative analysis, is there any evidence that horizontal gene transfer is playing a role?
Yes, because I didn't talk about horizontal gene transfer, although we've done a lot of work with horizontal gene transfer, but it's not really involved in virulence. So we see a lot of horizontal gene transfer with respect to secondary metabolite gene clusters. And so the answer is yes. It's like any other species complex. I guess when you track things, you do see some things showing up in one genome that is quite phylogenetically unrelated to another.
Question?
Yes.
Where is the canola seed increased for Eyre Peninsula, and how many years do they need to increase enough seed to plant Eyre Peninsula with, say, a new variety such as you did in 2012? Can they do that with a year's notice, or that's a very practical question?
I mean, seed increase is not an issue, if that's what you mean. It's not an issue for companies. It's more sort of the grower choice. So we were lucky that we were able to give advice in February, and growers make their decisions in February, March. So when that seed was, off the shelf, other seed was able to be sourced of other varieties. Is that what you're getting at? I missed it.
Well, I'm thinking if you want to switch varieties, it takes a couple years to increase to get enough seed.
Ah, true.
And also, where is the seed actually grown?
It's generally grown in places like Tasmania, where there isn't much commercial crops. We've had a lot of. If you worried about blackleg on the seed, also, is that an issue that you're trying to get at?
No, I'm just.
No?
Well,
okay.
I assume that the seed is not grown in Eyre Peninsula.
No, it's not. No.
It comes from Tasmania. Well, that would make sense.
Yeah.
Thank you.
Yes, it's grown in regions where there generally isn't many commercial crops. The question is, does secondary metabolites play a role in virulence? My group's worked on a secondary metabolite called cyrodesmin, which is closely related to gliotoxin, and gliotoxin's made by Aspergillus fumigatus. People who have aspergillosis, it's bad news. So we're very interested in secondary metabolite toxins. We've shown that if you delete the cyrodesmin gene cluster and the fungus cannot make cyrodesmin, you get a 50% reduction in stem cankering. So you get cotyledonary lesions, but the fungus, unable to make the cyrodesmin, is much more ineffective at stem cankering. However, we know that Leptosphaeria maculans lepidae, the very closely related one, can make cyrodesmin as well. We know that this gene cluster is not in any of the other biogloboses. And indeed, with these secondary metabolite toxins, indeed they are involved in virulence, but it's generally not in virulence to a plant. It's because they have a saprophytic stage of their life cycle in the soil. So the feeling is these days that it's generally a virulence factor in terms of all of the amoeba, bacteria, fungi, things in the soil that it's competing. But I didn't mention it in this talk because it, I felt that I didn't have enough time and it wasn't quite as related as the other stuff was.
Thank you very much, Dr. Howlett, for a very and I'll conclude by stating the obvious. It's clear from the work done by Barbara that the linkages between APS and MSA are really quite artificial because we're dealing oftentimes with one and the same. So thanks again.
Thank you so much.
For coming from very chilly Melbourne to Texas.
To a rather chilly ballroom, and yes, but it's great. Thank you.